Hey guys, so in this short lecture, I'll give a very quick overview of two methods from evolutionary genetics that can be used to test hypotheses in evolutionary medicine. So there are many different methods that can be used to test these hypotheses that we're going to cover in this class, but there's two with kind of outsized importance: selective sweeps and genome-wide association studies, also known as GWAS. So first up, selective sweeps. So a selective sweep describes the classical way of how we thought that a beneficial mutation increased in frequency to become fixed in the population, meaning every single individual in the population has the variant. Um, it might not be the only way that adaptation happens, but this is kind of the traditional way of thinking about how this occurs. And there's two important ideas about selective sweeps that are worth keeping in mind. First off, is that it's going to lead to a reduction or an elimination of genetic variation in the area around the mutation, the beneficial mutation. And then secondarily, positive selection can cause the nearby variants to kind of come along for the ride. So linked alleles in linkage disequilibrium with the beneficial alleles, can kind of hitchhike along and also rise to high frequency or even fixation. So I think it's useful to understand how precisely selective sweeps occur by walking through a little toy example. So here we have 6 sequences from a population, each line representing a different DNA sequence, and each dot, in this case, is a neutral mutation, meaning that it is neither beneficial nor deleterious. It's not really impacting the fitness of the organism. However, then along comes a beneficial mutation, so one that confers some kind of reproductive advantage to this individual, represented by the red dot on the second line. And then because that mutation shown in red confers some kind of fitness benefit to the individual, because that individual has higher fitness, that has more babies, and their babies have more babies, and so there's an increased frequency. The red allele increases in frequency in the population, and along for the ride comes variants nearby. So you can see that the blue-ish guy on the left and the hashed ones nearby are also rising in frequency because they're linked to the red allele. But then a recombination or crossing over event can break that linkage. So here you see a recombination event occurring in the upper left, and now the beneficial mutation in red is no longer always associated with the blue neutral mutation. So these guys are no longer, in this case, linked together. So ultimately, after many generations, the beneficial mutation is beneficial enough that it becomes fixed in the population, meaning everyone has it. And there's also, because those other neutral variants came along for the ride, nearby there's no or very little genetic variation in the population at this location, and for a region around the genomic region around it, everyone looks exactly the same. So in this little toy example, 3 neutral mutations have hitchhiked along and reached high frequency along with the red beneficial mutation. So what's cool is we can actually use various population genetic statistics to detect that these sweeps have occurred back in the past. So on your left, you have a scenario where there was no sweep, and in the right column, you have a scenario where there was indeed a sweep, using the same toy example. And then there's 2 kind of cartoon stats on the bottom. The top one is differentiation to other populations. So for instance, on the left, that leftmost population without the selective sweep is almost indistinguishable from any other population that's related that didn't have a sweep. But however, on the right, because that sweep occurred in this one population, it's quite different from another population. So the level of differentiation is high. On the bottom, another stat that's actually extremely powerful that we can use is to look at haplotype length. So haplotype length kind of measures how many linked alleles are being inherited together. So on the situation on the left, there's low levels of, short haplotypes because there's been no selection, so things are just evolving neutrally and there's plenty of variation. However, on the right, where the selective sweep has occurred, that sweep has brought along for the ride a bunch of hitchhiking neutral alleles, so you have these long haplotypes in the population. And so using these 2 stats, differentiation and haplotype length, you can often detect that a selective event occurred in the past. And in this way, you can actually test hypotheses about selection occurring in an organism's evolutionary history. So yeah, it's kind of the case that natural selection, the selective sweeps kind of cause these kind of massive scars in the genome that you can pick up on and be able to recognize that somewhere in that scarred region, there must have been some beneficial mutation that occurred that reduced variation and increased haplotype length at this location. This is another way of viewing that same idea. It gives the names of the population genetic stats that are used. So the differentiation stat that's most commonly used is called FST. And you'll see on the left it's low before the selection and higher after the selection. And then the second row gives haplotype length, in this case summed up by the stat across population extended haplotype homozygosity stat, which is XPEHH if you're looking for the abbreviation. But anyway, long story short, the thing to remember is that increased differentiation, meaning increased difference from and distinction from nearby populations, and long haplotypes are both strong signs that past selective sweep has occurred. So next up, for our methods that we're talking about today in evolutionary genetics, are genome-wide association studies, also known as GWASs. So the genome-wide association study is an observational study where you look to see in a population if any genetic variants that are present in the population are associated with a trait. Typically has to use a large group of individuals to have any statistical power. And quite often they're used to understand the genetic underpinnings of particular diseases. And since there's tons of different variants tested, and there's often what's called population structure, false positives are a major concern, and the hardest part about GWAS is understanding what's a real signature and what's actually a spurious false positive, because you're just testing so many times, you're bound to get some that by happenstance just associate with the trait of interest. So here we have a schematic of how GWAS works. So you look across the genome and measure or read some kind of sequence variation. This might be whole genomes, might be whole exomes. Quite often it's done with SNP genotyping arrays that allow you to read SNPs, or single nucleotide polymorphisms. And then you sequence this in two different cohorts. On your left, you see a bunch of people that are super stoked to have some kind of disease in this cartoon, and then on the right you have unaffected cohorts, unaffected cohort as a control. And then you look across the genome and try to find variants that are more often than not in the affected cases and not as prevalent in the unaffected controls. And then you can kind of guess that variant might be linked to the trait of interest. So you'll see on the bottom, this is what's called a Manhattan plot, typically because there's often more than one spike, and so it often looks like the Manhattan skyline. And shown on the y-axis is a p-value for the strength of the association of this variant with the trait of interest. And it's plotted on a negative log scale, so things that are higher up in value actually represent a smaller p-value, meaning they're more robust evidence that they're associated, this association between the genetic variant and the trait. So here's an example from one of the readings. On your left, you see that same GWAS value that we just talked about, indicating the strength of association of different variants for, in this case, kidney disease in African Americans. And you'll see that there's one particular variant is quite high. That's right over this gene, apolipoprotein L1. So there's some evidence that there might be association. And then on the right, the first method that we talked about, the selective sweeps, this is looking at haplotype length in Yorubans, who are a West African population. So in the what's called the wild type, although that's kind of a weird thing to refer to for humans, in black, the haplotype length is quite short, whereas the two reddish lines indicate that the haplotype is much fatter, much longer. That indicates that the allele G1 and allele G2 have some evidence of recent positive selection. So these two kind of lines of evidence together suggest that not only is this variant potentially associated with kidney disease in African Americans, but also that it was probably under selection in the ancestral West African population that a lot of black Americans came from due to the forced migration of the transatlantic slave trade. So anyway, those are our two methods, genome-wide association studies on the left and selective sweeps on the right, and just kind of an illustration of how they can be used to test evolutionary hypotheses relevant to medicine.